Hello students, I am Dr. Jaya Philip, Assistant Professor in the Department of Microbiology, Patna Women's College. In this lecture, we will study the basic principles of how cells communicate with one another, known as cell signaling. We will also discuss the different categories of cell signaling. Cells are not just simple building blocks, but they can detect what's going on around them and respond in real time to signals from their neighbors. This is called cell signaling. At this very moment, your cells are sending and receiving millions of messages in the form of chemical signaling molecules. These chemical signals are often secreted from the cell and released into the intracellular space. Not all cells can hear a particular chemical message. In order to detect a signal, that is, to be a target cell, a neighbor cell must have the right receptor for that signal. When a signaling molecule binds to its receptors, it alters the shape or activity of the receptor, triggering a change inside the cell. Signaling molecules are often called ligands, a general term for molecules that bind specifically to other molecules like receptors. The message carried by a ligand is often relayed through a chain of chemical messengers inside the cell. Ultimately, it leads to a change in the cell, such as alteration in the activity of a gene or even the induction of a whole process, such as cell division. Thus, the original intercellular signal is converted into an intracellular signal that triggers a response. However, not all sending and receiving cells are next door neighbors, nor do all cells pair exchange signals in the same way. So, based on this difference, there are four basic categories of cell signaling found in multicellular organism, that is, paracrine signaling, autocrine signaling, endocrine signaling, and signaling by direct contact. The main difference between the different categories of signaling is the distance that the signal travels through the organism to reach the target cell. Now let's discuss the category one by one. So the first one is the paracrine signaling. Often cells that are near one another communicate through the release of chemical messengers. This type of signaling, in which cells communicate over relatively short distances, is known as paracrine signaling. Paracrine signaling allows cells to locally coordinate activities with their neighbors. Although they are seen in many different tissues, paracrine signals are especially important during development, when they allow one group of cells to tell a neighboring group of cells what cellular identity to take on. One unique example of paracrine signaling is synaptic signaling, in which nerve cells transmit signals. This process is named for the synapse, the junction between two nerve cells where signal transmission occurs. When the sending neurons fires an electric impulse, it moves rapidly through the cell, traveling down a long fiber extension called an axon. When the impulse reaches the synapse, it triggers the release of a ligand called neurotransmitters, which quickly cross the small gap between the nerve cells. When the neurotransmitters arrive at the receiving cell, they bind to receptors and cause a chemical change inside of the cell, often opening ion channels and changing the electric potential across the membrane. Next is the autocrine signaling. In this type of signaling, a cell signals to itself, releasing a ligand that binds to receptor on its own surface. This may seem like an odd thing for a cell to do, but autocrine signaling plays an important role in many processes. For instance, autocrine signaling is important during development, helping cells take on and reinforce their correct identity. From a medical standpoint, autocrine signaling is important in cancer and is thought to play a key role in metastasis, which is the spread of cancer from its original site to other parts of the body. In many cases, a signal may have both autocrine and paracrine effects, binding to the sending cells as well as other similar cells in the area. Now the next is the endocrine signaling. When cells need to transmit signals over long distances, they often use the circulatory system as a distribution network for the messages they send. In long distance endocrine signaling, signals are produced by specialized cells and released into the bloodstream, which carries them to the target cells in distant part of the body. Signals that are produced in one part of the body and travel through the circulation to reach far away targets are known as hormones. Each endocrine gland releases one or more types of hormones, many of which are master regulators of development and physiology. And the last type is through cell and cell contact. Gap junctions in animals and plasmodesmata in plants are tiny channels that directly connect neighboring cells. These water-filled channels allow small signaling molecules called intracellular mediators to diffuse between the two cells. Small molecules such as calcium ions are able to move between the cells, but large molecules like proteins and DNA cannot fit through the channel without special assistance. The transfer of signaling molecules transmits the current state of one cell to its neighbor. This allows a group of cells to coordinate their response to a signal that only one of them may have received. In plants, there are plasmodesmata between almost all cells, making the entire plant into one giant network. In another form of direct signaling, two cells may bind to one another because they carry complementary proteins of their surfaces. When the proteins bind to one another, this interaction changes the shape of one or both the proteins, transmitting a signal. This kind of signaling is especially important in the immune system, where immune cells use cell surface markers to recognize the body's own cells and the cells infected by pathogens. So, in this lecture, we studied how cells communicate with each other and what are the different methods of cell signaling. I hope you found this lecture informative. Thank you.